Normally, a diploid germ cell will reproduce by dividing once to produce two daughter cells, which will each divide again in meiosis II for a total of 4 haploid daughter cells containing half of the chromosomal material, 23 chromosomes each. But sometimes it doesn't work that way. Let me show you a few pictures. During meiosis I and II, normally one diploid parent cell with 46 chromosomes divides twice and forms 4 daughter cells with 23 chromosomes each. For those with testes, these 4 cells are sperm. For those with ovaries, one of the cells becomes an egg and the other 3 are polar bodies, which nourish and create stability for the soon-to-be embryo. Did that make sense? Hope so, because it's about to get funky. Gene mutation is a change in the DNA sequence involving any number of base pairs, from changing one to altering large segments by deletion, insertion, repetition, or duplication. Extra chromosomes may be counted, or entire chromosomes may go missing. DNA unzips in the middle, then replicates into 2 by the enzyme helicase. Another enzyme holds the strands apart. Another, DNA polymerase, adds the new letters. DNA is transcribed into messenger RNA, which is translated into proteins. It sounds like quite a complicated mess. Any alteration of DNA is perpetuated and changes creation's greatest experiment, a human baby. More pictures! During meiosis, one way error can occur is misalignment of repeated sequences. Crossing over can occur unequally in a sperm or oocyte, creating a zygote with an extra or missing gene. A couple of bases by chance insert into newly forming DNA, which goes on to replicate with this error. Like a recipe, you can make changes, and though minor, the mutation will change the outcome. Some changes are simple polymorphisms and are common. Other mutations cause rare genetic conditions. Many pregnancies will miscarry where there are such mutations. 15% according to ACOG, and 60% of first trimester miscarriages are associated with chromosomal abnormalities. Let's talk a few definitions. Polyploidy is the presence of extra chromosome sets. This can happen if 2 sperm fertilize an egg, or a sperm fertilizes a diploid egg. If a baby survives, there are defects in nearly all organs, and it only lives a few days. Aneuploidy is an abnormal number of chromosomes. Rather than 23 pairs, there is either a deletion or addition of a chromosome. 2 types of aneuploidy are monosomy and trisomy. Most aneuploidy related to non-sex chromosomes leads to spontaneous abortion. Where the aneuploidy affects sex chromosomes, the symptoms are less severe, though brain and other development is impaired significantly. Monosomy, here a single chromosome is found where there should be a pair. If this is in a non-sex chromosome, it is almost always lethal. An example of monosomy is Turner syndrome, where there is only one X and no Y. Trisomy is a condition where there are 3 chromosomes where there should only be 2 normally. An example of trisomy is Down syndrome, trisomy 21. Normally, genetic males have the sex chromosomes XY and genetic females XX. Some syndromes where there is non-disjunction of the sex chromosomes include Klinefelter's syndrome, genetic male with an extra X, and XYY syndrome, and Turner's, genetic female with a single X. All of these will not survive, except the Turner's example of a single X. If a genetic female has an extra X, as in XXX, there is no disorder. You just have like a jug of wine from the Looney Tunes from 1946. Non-sex chromosome disorders are usually fatal, but some do lead to birth. Trisomy 21, Down syndrome, trisomy 13, Patau syndrome, and trisomy 18, Edwards syndrome are examples of a 3rd chromosome condition. Rarely carried to term are also trisomies on chromosome 15, chromosome 16, and 22. Deletion is where base pairs of DNA are removed outright, leading to phenotype alterations such as genetic male infertility by deletions on the Y chromosome, or autism symptoms from microdeletions on chromosome 16. Causes of mutation come by 2 types of changes in DNA. Spontaneous mutations. These are some things that just go wrong, like strand slippage or bonding of strands that are imperfect. An example of point mutation is substitution, where a letter changes in the sequence. It can be repaired by proteins, which slice it out and replace it properly, but half the time they replace the wrong side. DNA can break apart by radiation or some environmental exposure. It can be repaired, but sometimes with errors, such as additional letters in the strand. There are larger mutations. We lose big portions of the chromosome, affecting hundreds or thousands of the genes. This is deletion. Duplication, where chromosomes have a repeated section, and inversion, where it flips instead of sequence lining right up, are also problems. Insertion, where part of the chromosome inserts to another chromosome, and translocation swaps between DNA strands. There are induced mutations. These come by environmental factors like radiation or cigarettes. In experiments, scientists can use radiation and other forms of X-ray, liquefied cigarette smoke, or other things to induce mutations, delete a few bases or break chromosomes, whatever they need to have happen. For example, scientists can use liquefied cigarette smoke, which breaks chromosomes, which then bind together on other strands, activating cancer-causing genes. That's crazy. Common products that contain mutagens are hair dye, smoked meats, certain flame retardants used in children's sleepwear, and food additives. As you can see, there are many ways where genes can get messed up. The war just introduced interesting variety into the human species. So do your homework before you make a baby.